Welcome to This Explainer. Today, we're going to dive into something really wild, the fascinating chemical language that allows microbes to talk directly to your cells. We are exploring the absolute cutting edge of biology to understand how the gut microbiome essentially hacks our cellular receivers, an expansive network known as the GPCRome.
We constantly hear about how the microbiome impacts our health. Eat your fiber, take your probiotics, right? But here's the real mystery. How do these microscopic bacteria actually send physical messages to human tissue? How are they communicating with our bodies on a molecular level to trigger such massive changes in our physiology?
Well, to give you an idea of the scale we're talking about, just look at this number, 500,000. Our human metabolomic profile contains at least 500,000 distinct metabolites. And here is the absolute kicker. The vast majority of these molecules aren't even of human origin. They're actually produced or modified by the trillions of microbes living inside us, churning out this massive chemical vocabulary that we are just now beginning to translate. So, here's our agenda for today. We're going to cover 1. The microbiome's missing link. 2. Meet the host GPCRome. 3. The art of chemical mimicry. 4. Screening the bacterial metabolome. 5. The gut immune-brain axis. And finally, 6. Unlocking future therapies. All right, let's jump straight into part 1, the microbiome's missing link, the vast chemical unknown.
Okay, let's start by zooming our mental camera all the way down to the biological intersection where all this communication goes down, the intestinal wall. It's this incredibly bustling, dynamic hub where your diet, your host molecules, and microbial molecules all mix together. This isn't just some passive digestive tube. It's a highly active interface where microbial specialized metabolites cross over to regulate our systemic immune function, influence our brain activity, and even affect our muscle health.
Moving on to part 2. Meet the host GPCRome, the cellular locks.
Let's zoom that mental camera in even further, right down to the surface of our human cells. This is where we find the crucial receivers of these signals, GPCRs, or G-protein-coupled receptors. This is literally the largest class of human membrane receptors. You can think of GPCRs like millions of intricate molecular locks lining the outside of your cells. Their entire job is to sense extracellular molecules and trigger downstream physiological responses inside the cell. They're just sitting there, waiting for the exact right chemical keys to turn them on.
Now, for the longest time, science assumed that only our own endogenous human ligands, our internal signaling molecules, were the only keys that fit these locks. But guess what? It turns out bacterial metabolites, these small molecules produced by gut microbes, can also perfectly fit these human locks. They just sneak right in and turn them, initiating cellular cascades exactly like our own biology would. Which brings us to section 3, the art of chemical mimicry, master impressionists. Just look at this striking resemblance here. On the left, you've got bacterial N-acyl amides, and on the right, human endogenous GPCR ligands. Structurally, they are practically identical. The bacteria are producing precise structural copies of our own human lipid messengers. It's just a brilliant evolutionary adaptation to make sure their signals are heard loud and clear by our bodies. So the key concept to grasp here is chemical mimicry. By crafting these molecular counterfeits, commensal bacteria can directly bind to host receptors, like GPR119, which regulates your glucose and metabolism, and G2A, which is super involved in immunity. They are essentially hijacking our native systems. Well, hijacking may be a strong word, but they're definitely taking the wheel to control human physiology. All right, part 4, screening the bacterial metabolome, finding the keys.
So you might be wondering, how did researchers actually figure out which of these bacterial keys fit into our human locks? Well, they went through a massive bioassay-guided isolation process in 3 pretty intense steps. First, they cultured individual bacterial strains from the human gut. Second, they extracted the secreted metabolites from those cultures. And third, they systematically screened those metabolite libraries against 241 different human GPCR targets.
Basically, researchers managed to map the chaotic soup of human-associated microbial outputs directly to specific cellular receptors. They took these extracted metabolite libraries, dropped them into engineered cell lines, and then literally watched to see which human GPCRs lit up in response to the bacterial signals. And wow, the results were absolutely stunning. This heat map reveals the specific human GPCRs activated by distinct bacterial strains. See those dark blue squares? That indicates strong agonist activity, meaning the bacteria successfully turned the cellular lock on. It proves for sure that specific bacterial strains light up very specific human receptors. In some cases, this process actually deorphanized GPCRs, which is just a fancy way of saying it found the functional keys for receptors whose purposes were previously completely unknown to science. Next up, Section 5, the gut-immune-brain axis, systemic impact.
Because these microbial molecules act as keys for our GPCRs, they trigger sweeping physiological changes throughout your body. We are talking massive impacts here. Improved glucose homeostasis, the trafficking of immune cells for tissue repair, the secretion of vital endocrine hormones like GLP-1, and get this, even modulating the permeability of the blood-brain barrier.
Essentially, what this means is that these seemingly simple commensal bacteria floating around in our gut aren't just passive little passengers helping us digest food. No way. They are active systemic effectors, constantly pulling the molecular strings of our immune and neurological systems, acting as a crucial driving force in the whole gut-brain axis. Finally, Section 6, unlocking future therapies, microbiome biosynthetic gene therapy. This research points us toward a really profound conclusion. Manipulation of microbiota genes encoding metabolites that elicit host cellular responses represents a possible small molecule therapeutic modality. Simply put, if we can map these bacterial effectors, we could eventually deploy targeted microbiome biosynthetic gene therapies. Imagine treating metabolic, inflammatory, and neuropsychiatric diseases by simply tweaking the microbial factories naturally living inside us, rather than relying on traditional systemic drugs. All of this leaves us with a deeply fascinating and honestly kind of provocative thought to walk away with today. If bacteria literally possess the molecular keys to our cellular locks and can dictate our immunity, our metabolism, and even our brain function, who is really in control? Who is actually driving our biology? Thank you so much for joining me for this explainer. I hope this deep dive into the GPCRM and our molecular translators has completely changed the way you view the microscopic world inside you. Keep questioning and keep learning.